so maybe 5 minutes after lecture, I'll just show you some ways to get some reliable sources for your poster. But let's start with a little bit of review from animal behavior. So remember the honeybee waggle dance? So diagram that dance for you and note what information each part of the dance conveys to the watching bee. Right. The waggle dance conveys two important pieces of information about a food source. Up. Hopefully this was too difficult to draw a bee. I do not have any artistic skills whatsoever. I ended up drawing like a stick figure bee. But that's okay. That's okay. As long as you get the point across, right? There's only one Rembrandt. It's inside. So remember the honeybee waggle dance. So there are two parts of the dance that convey two pieces of information. So the angle of the dance is relative to the vertical, right? Because most honeycombs are vertical, is the direction of the food source in relation to the sun. Bees navigate by the sun. And so if the straight part of the waggle dance is straight up, that means the food source is straight towards the sun. If it's 30 degrees to the right, then it tells the bees to go 30 degrees to the right of the sun, right? And it shapes a figure 8 like that. So the direction of the straight run is one piece of information, and then the waggle, right? The duration of the waggle indicates distance, and a rough approximation is 1 second waggling is about a kilometer of distance. But all you really need to know is the longer the waggle, the further away the food source is. The bees know the route, so that's probably cool. So it's actually fairly sophisticated, communicating fairly sophisticated information for a bunch of insects. So, so any questions about the waggle dance? Okay. So sometimes I might ask you to think about, hypothesize about an experiment to demonstrate some particular piece of information that we have learned. So draw a setup for an experiment designed to show that solitary wasps locate their nests by spatial learning. So remember what spatial learning is. We talked about how might you test that in solitary wasps who leave their nest and come back. How do they know how to get to the nest, and how might you demonstrate how they figure that out? finish up. So here's what the setup looks like. Remember, spatial learning, animals locating objects by learning the relative distances from other objects. And this was an experiment that was done with solitary ground nesting bees. So an experimenter put a set of objects, in this case pine cones, around the nest and left them there for several days so that the bee could use them as a navigation cue to get to the nest. Then one day, when the poor bee was out foraging, the mean experimenter moved all of the pine cones 6 feet to the right so that they surrounded a place that the nest hole was not in the middle. And the bee returned to the middle of the pine cones rather than to the nest. So that suggests that the bees navigate by spatial learning only, right? There's not like there's maybe a hormone or a scent that comes from the nest that they can use. Now this is a pretty brute force type of experiment. What might you do to maybe make this a little bit more subtle of an experiment? To learn maybe some more subtle things about the bees' spatial learning. What might you do instead of just taking everything and shifting it 6 feet to the left, to the right on one particular day? What might you try?
Could you change the object that you put around the nest?
Good question. Yeah, I was thinking about that, right? What if you changed them to rocks while the bee was away? Would the bee still know that the circle of rocks was basically the same as the circle of pine cones and the nest would be in the middle? Or would it get completely freaked out because the pine cones are gone? Good question. That would be an interesting experiment, right? What else might you do?
Like maybe different scents?
You could try different scents. Yeah, that's true. Although in general, it seems like, at least with this species of bee, that it's entirely using spatial learning. But yeah, maybe you could like, instead of shifting it 6 feet to the right all in one go, what if you move the circle maybe 6 inches to the right every day, right? So slowly the nest was moving to the edge of the circle. Would the bee respond to that? Is it enough that they can notice subtle changes every day? Or is it kind of an all or nothing, where it's either the center of the circle or the bee will be lost? So there's some interesting experiments, things you can do. And sometimes I might ask you, hey, what is an experiment, maybe on a question, maybe it's extra credit, right? What's an experiment you might think of to learn more about this system? And it's entirely an open-ended question. I don't have usually an answer in my mind, so come up with something. Use your originality, use your imagination. And there are very few experiments that are bad experiments. Some may not be feasible, or in some cases, we may know enough to know that that experiment wouldn't give us the result that we desire, but that's okay. But being able to design experiments is an important part of being a scientist, or even being just a regular person, right? I tell my students in my UCS 101, right, who are not all scientists or not all science-minded, but we use the scientific method every day. We experiment on things every day. And so, even if you're not going to be a scientist for a career, I think understanding the scientific method, how we proceed by, hypothesis, experiment, observation, is important to be able to do things in your life that you might not ordinarily do. So, Okay, so any questions about the animal behavior stuff that we covered? So if not, we're going to talk about plant communication today. And yes, plants do communicate. So plants can communicate in a number of different ways. Plants can communicate internally, right? So different parts of the plant talk to other parts of the plant. The leaves talk to the roots talk to the stem, things like that. That's not unusual, right? All different parts of our body communicate with each other as well. So plants can communicate in terms of growth, right? Make sure that the stem is not too tall for the root system to support, right? If the root system has to grow more, slow down the growth of the stem for a while so the plant is more likely to get blown over, right? Especially for your tree. Tropism. You guys know what a tropism is? So a tropism in biology is basically an attraction. So plants are attracted or repelled by different things. So for example, light, right? Plants photosynthesize, they need sunlight to grow. So seedlings grow up towards the light. They display an attraction to light. It's called phototropism. Seedlings also respond or show a tropism to gravity, right? If you're a seedling and you're buried in the ground, you don't know which way is up, but seedlings can sense which way gravity is going. And they know that growing against gravity is growing up. And eventually they'll break through the soil and they'll come to the sunlight. They took seeds up on the space shuttle, right, where there's no gravity. And those poor seeds got completely freaked out. They didn't know what was going on. They grew in like little circles trying to find gravity so that they could grow upwards, but there was no gravity. Plants can communicate stress, right? So if a plant if there's a drought or excessive heat or excessive salt in the soil, right? The roots can communicate with the leaves. The plant can slow its growth if there's a drought and there isn't enough water. The plant can activate certain mechanisms. So, for example, some soils have a lot of salt, or some of them are contaminated with heavy metals, things like that. Some plants can change their biochemistry so that either A, they're not as affected by the extra salt, or B, they have ways to remove that extra salt from their bodies, from themselves. So that plants can do a whole lot of internal communication, but plants can also do more like what we would think of as communication, external communication, where one plant communicates with another plant. And we'll talk about plants can communicate the presence of predators, right? There are lots of animals that eat plants. Think of, the gypsy moth caterpillars in the spring, right? They hatch and they go all over and they chew up all the leaves on the plant. Plants can communicate with each other saying, hey, I'm being attacked by caterpillars. You better turn on your chemical defenses. Plants have chemical defenses to drive those away. Same thing with diseases, right? Disease attacks one plant in an area, that plant will signal to other plants that it's diseased, and the other plants can actually change their biochemistry to repel or beat off that pathogen. So plants can communicate with each other. So this is an example of phototropism. It's actually called photomorphogenesis because photo is light, right? What's morpho? Somebody morphin Power Rangers, come on guys. It's shape, it's more shape, right? And genesis, of course, is the beginning, the acquisition of it. So this is basically plants gaining shape from light. And this is from the lab of a man named Roger Hangarter at Indiana University. It's called Plants in Motion. So he has lots of really interesting kind of time-lapse pictures of corn seedlings growing and behaving. And he actually came to Hiram a few years ago to talk about this, and that's kind of where I got the idea to show some of these things. So what this video is going to show you is that this plant, this side, this is in total darkness. There is no light in this side. This side, there is light. And so what you're going to see is when plants detect light, they're going to start to spread their leaves out, right? And they're going to start to try to photosynthesize. They're going to change their shape. If there's no light, the plants don't know that they've broken through the surface yet. So they're just going to keep growing straight up. They're not going to put their leaves out because they don't think they're through the surface of the soil yet. So watch these guys. And this is just silent regeneration. So remember, this is total darkness. This is light. That's why this is black and white. See, these guys are already spreading out their leaves. These guys are like, hey, we're still under the ground. Where the hell's the light, dude? You keep growing up and up and up until eventually they fall over because they get too tall. So light can cause plants to change their shape as they encounter it. If they don't encounter it, they don't change their shape. So I talked a little bit about this, but this is a very interesting biochemical process that you guys are going to run up against again and again. And it's a phenomenon called signal transduction. So the cell membrane, right, the lipid bilayer, is impermeant to most things. And plants, in addition to a cell membrane, have a cell wall, which gives plant cells their rigidity. Right, that's why the tall stalk can support all those leaves, because plant cells are actually pretty strong in terms of being building materials. Problem is, a lot of things can't get from the outside of the cell to the inside. Like, for example, a hormone or a chemical signal that says, hey, I'm being attacked by a virus. We ought to change our biochemistry so that we're less likely to get attacked by the virus. So the process of signal transduction is the process whereby information that's outside the cell gets transmitted to the inside of the cell and can make the plant change its behavior or its biochemistry or something like that. Transduction means to carry across, to transduce something, to carry it across. So signal transduction is to carry the signal across the cell membrane from the outside to the inside. So there are 3 phases to this process. There's reception, transduction, and response. And there are 4 components, 4 physical components. The first one is the signal. Let's say it's a hormone. Or it could be something from the environment. Even light can be a signal. The signal binds to a receptor. So usually the receptor is on the cell surface. So it's exposed to the outside. So the signal can easily detect. Sometimes the receptors are intracellular and the signal passes through the membrane, but that's pretty rare. Most of the time the receptor is here in the plasma membrane and it sticks through it. So one part sticks out and one part sticks in. And so when the signal or the hormone or the stimulus binds to the receptor can carry that information to the inside of the cell, transduce it, and relay it to another set of chemicals that are called second messengers. These are small molecules. They diffuse very rapidly. Cyclic GMP is one example, or even ions like calcium can be second messengers. The receptor releases or activates these second messengers. They diffuse throughout the cytoplasm of the cell, and they induce the cell's response. And the response can be any number of, the cell can respond in any number of ways or sometimes multiple ways. So sometimes the response is a short-term change in cell physiology. It may change its biochemistry for a short period of time, or it might reinforce itself against water loss if there's a drought or something like that. But sometimes the response can be long-term. Sometimes the cell can change its genetics, change which genes are active and which genes are not, and this can make long-term changes in cell physiology or even in the plant structure itself. So the idea of signal transduction, the idea of a signal being bound to a receptor and relaying information through a second messenger and getting a response, is a very common one in biology, and not just in plants, in animals as well. We have lots of second messenger pathways where, say, a hormone binds to a receptor. Example might be growth hormone, right? Animals have growth hormones, plants do too, actually. Right? Growth hormones signal cells that they have to divide. It's time to grow, it's time to get bigger. So growth hormone is released into your bloodstream. It binds to its receptor, and growth hormone receptors are on almost every cell in your body. The receptor creates second messengers, and those go to the nucleus of the cell, and they activate genes that turn on the process of cell division. And so that's a growth signal. Right? You are pretty much growing through most of your childhood. Once you hit adolescence, right, your growth slows and stops. Your body doesn't produce as much growth hormone anymore. You've reached your adult size. Sometimes, though, people who suffer from certain types of diseases, particularly like tumors in their pituitary gland. The pituitary gland is responsible for releasing growth hormone. And so when you reach maturity, it slows down and stops. But sometimes people get tumors in their pituitary gland, and that wakes it up again, and it releases growth hormone, and people start growing again. So if you actually start growing again, it's actually not a good thing. It means there's probably a tumor on your pituitary gland. The tallest man in the world, who's currently alive, who I think is like 7'9" or something like that. That happened to him. So basically, he's a normal-sized man, developed a pituitary tumor. The pituitary gland woke up, released growth hormone. The cells received the growth hormone and started to grow again. And he started to grow again. Until, I guess hopefully they found the tumor and they were able to treat it. So second messengers are important in biology. You see them all the time. You're going to see them more in this course. If you go on to molecular cellular biology or animal physiology or genetics, you'll see them there too. So signal transduction cascades are very important. Here's a specific one in plants. Okay? So this is called the de-etiolation or greening response. So we saw in the movie, right, the plants were growing here and they were green, right? The leaves were green. The plants in the constant darkness were pale white, right? If you keep plants in the dark, they don't turn green. So de-etiolation or greening is mediated by light. And so in this case, in terms of signal transduction, the stimulus is not a hormone, but photons of light. Photons of light enter the cell, they cross the cell wall and the cell membrane. They bind to a protein called a phytochrome, which is activated by absorbing photons of light. Phytochrome produces a second messenger called cyclic GMP, and that activates a protein kinase. Protein kinases are enzymes that put a phosphate group on another protein, and that usually changes its activity. In this case, the protein is in the nucleus, and here's the DNA, and basically the protein changes which genes are active. It activates certain genes, including the genes for chlorophyll. Because chlorophyll is part of the photosynthetic pathway, which is how plants get their energy. It's also what gives plants their green color. There's also a second pathway, which the phytochrome activates calcium channels in the membrane of the cell. So much like sodium and potassium channels we learned about in neurons, there are channels that allow calcium to cross into the cell. And calcium also activates protein kinase enzyme, and that also alters gene expression. And so basically the plant starts to produce chlorophyll, starts to carry out photosynthesis, and turns green. So this is a whole signal transduction mechanism from reception to transduction to response, in response to photons of light. So your signal can be almost anything, from a chemical to a photon to even sometimes temperature changes can activate signal transduction pathways. So just keep that in mind. That's a pretty important thing. Plants have hormones, plant hormones, right? What are hormones? Well, in animals, hormones are small molecules that circulate in the blood. But plants don't have blood. So how do plants have hormones? And how do those hormones get to where they need to go? Right, basically plants have pathways, right? You guys remember xylem and phloem? We got that in high school biology. Basically there are things that are kind of like blood vessels in plants. They carry water and sugars and fluids, not blood. But also hormones travel up and down different parts of the plant. There are a number of plant hormones that are very important for plant survival. The major group of hormones are called auxins. Those are hormones that regulate growth. So plant auxins are associated with plant growth. And so a lot of people who are in agriculture, for example, are very interested in plant auxins. Right? Because if you can get your crop plants to grow faster or produce more fruit or more seeds, you're going to make a better profit. So there's a lot of study on plant auxins. There are a group of hormones called gibberellins, strange name, but what they do is they mobilize nutrients in seeds, right? So plants produce seeds. When the seeds fall or are dropped into the earth, right, they get buried and they start to sprout. The sprout needs energy to get to the surface and start photosynthesis. The seeds have nutrients in them, right? If you eat like nuts, right? Nuts are just seeds. So all the nutrients in those seeds get mobilized by these gibberellins when the seed gets buried, and then the plant starts to sprout. Grasnosteroids are similar to steroid hormones in humans, and they also affect growth. And there's also, interestingly enough, there's a gas. Ethylene is a gas. Ethylene is the gas that promotes ripening in fruit. It also promotes what's called leaf abscission. So when in autumn, when the leaves fall, that is due to an increase in ethylene produced by the plant, and it basically breaks down the joint between the stem, right, and the little place where the leaf is attached to the stem, and then the leaves fall. That's to do with ethylene. So, for example, right, a lot of times like at the supermarket, tomatoes particularly, right? Tomatoes are pretty fragile. So when tomatoes are picked, they're picked green because they can be shipped in big boxes and they don't collapse, right? Green tomatoes are pretty tough, but not too many people like to eat green tomatoes, at least not if they're not fried. So when they get to the grocery store, the grocer wants them to ripen and quickly. And so what they do is they basically have giant containers of ethylene gas. You put all the tomatoes in a big box, you seal it up, you turn on the ethylene gas, and in a couple of days they're nice and red and ripe. Leaf abscission, so if you, experimentally, if you go around with a tank of ethylene gas, and say you go up under a tree, and even if it's like midsummer or late spring, right, the leaves are out, if you release ethylene gas, they'll start to fall. even though fall is months away. So sometimes the signal or the hormone can be not a chemical or a liquid, but a gas, which is pretty interesting. So here's an interesting video, another one from Baumgartner's laboratory. So this is phototropism in corn shoots. So he has a single light, and the corn shoots sprout and they grow. And then as they grow, they start to lean towards the light. Plants show a phototropic response. They grow towards the light. And you've probably seen this, right, in maybe a house plant in your house or in your parents' house, right, where it sits, and if it's near a window, it'll grow towards the window. And eventually you can see the whole plant like leaning over to try to get to the window. And so you kind of turn it, right, and then the plant sort of changes direction and goes back to the window. So the phototropic response of plants is really, really strong. But this is kind of cute because what he does is he turns the light off, and then the plants lose their phototropic response, and then he turns it back on. And it kind of looks like they're worshiping the light. Little plants worshiping. So let's see how that looks. So this is phototropism in corn shoots. See, they're all leaning towards the light. Light goes off. Now they go straight up. They're not, the phototropic response isn't there. And turn the light back on, they'll go back to their phototropic response. So phototropism in plants is a very strong part of plant growth and plant development. So phototropism is not something that's new. People have known that plants bend towards the light for hundreds of years. But for a lot of the time, it was kind of, nobody really knew why, right? It was sort of accepted, this is what happens. Until the 1800s, when sort of the enlightened values of science and scientific experiment started to come up, and people started to wonder, well, maybe I can figure out why plants bend towards the light. And so some of the early phototropic studies were actually done by Charles Darwin, who's famous, of course, for the theory of evolution. But he was also an avid gardener, had a greenhouse, and so he grew plants, and he was interested in the phototropic response of plants. And so what Darwin observed, right, was that the seedlings that grew in his garden would orient their tips towards the light, whichever way the light was going. So Darwin hypothesized that maybe that response is only coming from part of the shoot, not the whole shoot. And so it's a really interesting sort of example of the scientific method in action, right? So Darwin first thought, well, maybe it's the tip. And so what Darwin did was he cut off the tip. And lo and behold, the plant did not show the phototropic response anymore. Right? So no tip equals no phototropism. Problem solved, right? But yeah, maybe not. Why not? What would you criticize about this first experiment that Darwin did? Lock the tip off, no phototropism, clearly the tip is responsive. But what are some problems? Ray, what do you think? What are some potential problems with that experiment?
Where are the resources going? Is it going to regrow that tip, or is it just ignoring the phototropism?
excellent question, right? Maybe the plant just is too busy fixing the damage to display phototropism, or maybe the damage itself caused the plant to stop moving or to maybe even die. So, right, cutting off the tip of the plant is pretty drastic. So what Darwin decided to do is, okay, let's go a little bit further. So instead of cutting off the tip, he covered it with a little cap. And the cap was opaque, so no light could get through. And lo and behold, again, the shoot did not display the phototropic response. So in this case, the cap, right, showed no phototropism. So it wasn't just cutting off the tip. It wasn't the damage that he did to the plant that led to the lack of phototropism. It was something about light getting to the tip. But what's another concern? What might you say to Darwin about the second experiment? It's not exactly proof because something. Right? What if you were just standing around and someone put a big bucket over your head? Would you start walking around so you would bump into things? No, right? You just kind of stand there, wait for someone to take the bucket off. So if someone put a bucket on your head, they might just think, well, humans with buckets on their head don't move, right? But that's not true. You can move, you just choose not to move. If somebody put a bucket on your head that was clear so you could see, you'd probably walk around. So that was the same type of criticism Darwin got for the second experiment. People said, well, what if the cap is just too much of a problem, right? We're talking about a shoot, so it's a very small plant. What if the cap is too heavy and it won't bend? So you're not blocking the phototropic response, you're just burdening the plant to the point where it can't behave, can't react. So Darwin said, okay, dude, so what I'm gonna do is I'm gonna make another cap, but it's transparent, so light can get through. Aha! In that case, it does show phototropism. So it clearly isn't the presence of the cap or the weight of the cap. It's the fact that light can't get there. So opaque cap, no phototropism, transparent cap, phototropism. So basically, three very simple experiments. Darwin came up with a very very well-tested conclusion, right? He knew it was light on the tip. That was what was happening. He actually did a couple of other experiments. So for example, someone said, well, what if it isn't just the tip? What if it's the whole thing? And so he covered the bottom part of the shoot instead of the tip, and it still showed phototropism. But nobody really thought that was, it was just kind of him making sure that all the I's were dotted and T's were crossed. So Darwin's experiment showed, yes, the tip of the shoot is responsible for phototropism. But that doesn't really answer the whole question, right? Why do the plants display phototropism? It's not magic. There's something going on inside the plant. But in the 1850s, Darwin didn't have access to all the modern tools of biochemistry. So he didn't know, he couldn't figure out what was going on with the tip. But another important aspect of the scientific method, right, is not only do you do the experiments, but you write them down and you communicate them so that other people can do more experiments. And if they can't do it in 1850, maybe they can do it in the 1900s, right? Maybe when technology is advanced enough. And so Darwin did these experiments and kind of left those conclusions out. And eventually, when the science caught up to what people could do, other people took this and said, okay, now we can figure out what is it that's going on in this tip of the shoot that's causing phototropism. So in 1913, a scientist by the name of Peter Boysen-Jensen took Darwin's experiments, he read the papers that Darwin had published, that talked about the tip being responsible for phototropism. And he said, okay, but what is it? So let's say it's something that comes from the shoot and goes up into the tip. What happens if I could block the movement of whatever that thing is inside the plant? And so what he did was he took a barrier, basically, and he cut the tip of the plant off, he put the very thin barrier in between, and then he put the tip back on. Now, if the barrier was made with gelatin, which is permeable to liquids, the plant still showed the phototropic response. But if you replace that with a little piece of mica, right, which is a very thin rocks, probably played with mica when you were a kid, but it's impermeable to water, so liquids can't cross. When we did that, the plant did not show the phototropic response. So Darwin showed it was the tip. Boyes and Jensen added the next thing, which was that it was something liquid that was coming up from the bottom of the plant, and if it could get to the tip, the plant would show the phototropic response, but if it couldn't, it wouldn't. Okay, interesting, but still, what is that thing? What is that thing that's inside the plant? So again, a few decades later, you get a little bit more sophisticated. So Boyes and Jensen showed that the signal was some diffusible molecule. Okay, then what is the diffusible molecule? So Fritz Wendt in 1928, so another about 15 years after Boyes and Jensen, showed that this signal that comes from the bottom of the plant, the way it works is to elongate cells on the side away from the light. So basically phototropism is not so much growth towards the light, it's growth on the side away from the light. So, right, the plant cells are nice, kind of even squares for the most part, right? And when they're roughly equal size, the plant grows straight up. Let's say light comes in from this side. Wendt shows that in that case, the side away from the light, these cells start growing bigger. And right, if these cells start growing bigger and these don't grow, that's going to push the whole thing over to the right. Right, because these cells are not going to grow, but these ones are. They're going to grow longer, and so they're going to push the whole thing over to the right. And so this is how Wendt did that experiment. So he knew Boyes and Jensen used this little agar cube, and he showed that whatever it was could pass through the agar cube. Well, Wendt said, okay, well then the agar cube must have this thing in it, right, if it passed through it. So what he did was he took a tip, and he put it on the agar cube, and whatever chemical promoted this, this growth on the side away from the light would diffuse into the agar cube. When you put the agar cube straight on the top, it would grow taller, but it wouldn't bend. It wouldn't show the phototropic response. But if you put the agar cube on one side of the plant, one side of the shoot, it would bend the opposite direction. So if you put the cube on the right, it would bend to the left. If you put the cube on the left, it would bend to the right. Even in the absence of light. So here he said, okay, whatever the substance is, it causes the plant to grow. And if it's only on one side, it causes more growth on that side, which means the plant leans the opposite direction. Okay, so the signal elongates cells on the side away from the light. So basically 3 sets of experiments over a period of about 75 years got us from plants bend towards the light from the tip to there's a hormone that, or there's something in there that makes the plant cells grow. So it's a growth hormone or growth, whatever it is. And it works on the side away from the light. And that, that growth hormone actually turned out to be this plant growth hormone auxin. auxin. So the plant cells on the side close to the light decrease the amount of auxin they produce. The cells on the side away from the light increase the amount of auxin they produce. And so that causes that differential growth, which bends the cell towards the light. And that was actually the first study of plant growth hormones. They discovered auxin, and they discovered a whole bunch of other plant hormones that affect growth. And that was kind of a big step forward in terms of studying plant growth. So, just kind of an interesting illustration of how the scientific method works. Right? Somebody starts one idea, simple set of experiments, show one small fact, it's the tip. Boyes and Jensen's experiments showed it's not just that it's the tip, but it's something diffusible, something that can move through an agar piece of gel that's water permeable, and presumably anything that dissolves in the water, but it can't cross the mica, which isn't water permeable. And then Wentz showed that that substance, whatever it is, causes growth, and if it's offset from the center line, it causes growth on only one side, which pushes the shoot to the other side. So clearly the light causes the increase in auxin on the opposite side, and that's how you can vote. So very interesting way to kind of take these gradual steps until you make the big discovery. And that's how a lot of science works. It's not usually one person makes the huge discovery, right? They're building on what people have done before. And maybe the stuff before didn't seem so spectacular, but when you actually get down to the nitty gritty and you discover the cause, you discover the plant hormone auxin, which has been very, very fruitful in studying plant growth and particularly ripe crops and things like that. So, so a really interesting kind of illustration of how science really works. So remember I also talked about communication outside the plant. And there's actually been a lot of work on plant communication, communication between plants in the last 10, 15 years. So actually this slide is really, this used to be kind of all of what we knew, and now it's really just a tip of what we know. But we know that plants can communicate with each other. They use chemical signals that diffuse through the air, so similar to pheromones. And they can communicate things like environmental stressors like drought. They can communicate about the presence of herbivores, damage to an individual plant, right? The plant can send signals to other plants in the area so they can maybe affect a response that will help reduce or eliminate the herbivore's damage to the plant. And of course, pathogens. So here's an example of a herbivore causing damage to a plant. This is actually a really interesting response because it uses basically one very interesting biological fact that somehow the plant has figured out. So there is a particular caterpillar that grows on or lives off of the leaves of a particular plant, right? The caterpillar is the larval stage of a moth or a butterfly. It eats the leaves until it grows big enough, it pupates, and then it turns into a butterfly or moth and flies away. So this particular caterpillar, the plant that it's living off of, right, doesn't want to be food. So interestingly, a combination of chemicals, one that comes from the damaged plant cells, and one that comes from a chemical in the saliva of the larva, the caterpillar, combine and cause the cells in the plant, they activate a signal transduction pathway. And the plant produces volatile chemicals that go out to the air, pheromones. And this particular pheromone attracts one particular species of parasitoid wasps. I don't know if you're familiar with these parasitoid wasps. There are lots of videos of them on YouTube and stuff. It's kind of disturbing. But what they do is they generally parasitoid wasps are very specific to the type of host. Usually it's a caterpillar. They sting the caterpillar, they paralyze it, they lay their eggs on it, and then when the eggs hatch, the baby wasps eat the caterpillar for food. Well, this particular parasitoid wasp is a particular parasite on this particular species of caterpillar. So the plant is basically calling these wasps saying, hey, there are a whole bunch of these caterpillars eating my leaves. Would you please stab them, paralyze them, and then lay your eggs so that your offspring can eat them? So they recruit these parasitoid wasps, they come in, and they do exactly that. They paralyze the caterpillar, they lay their eggs on it, the caterpillar larvae, the wasp larvae eat the caterpillar, no more threat to the plant. Right? It's almost like ordering a hit on somebody, kind of, right? It's like, hey, come over here. I know you do that. I know you can kill this guy. Why don't you do that? So very interesting pathway and very specific. I don't know actually if all plants. Some plants may do this. Because the wasp and the caterpillar usually have a very close relationship, a wasp that parasitizes one species of caterpillar may not parasitize a very closely related species. So it may be that this response is very specific, or it may be that only when this particular species of caterpillar is involved does the wasp actually do the job that the plant wants it to. But either way, it's a very interesting type of plant communication. Now, plants communicate also when there are pathogens, right, diseases present. Pathogens are generally classified as either virulent or avirulent. Avirulent pathogens are not particularly dangerous or damaging. We carry a lot of bacteria and viruses in our bodies that don't cause disease. They just kind of go along for the ride. In some cases, our biochemistry has evolved along with these bacteria. So avirulent pathogens are generally not a problem. But virulent pathogens which cause damage are a problem. So there are a couple ways that plants can battle infection, just like our immune system can battle infection. One way is called a hypersensitive response. And what a plant can do is, if there's a small region of infection, it can actually wall that region off to prevent the pathogen from getting access to the rest of the plant. So you go like walking around the field station or something, you ever see those giant blobs on the side of trees, bolls, right? So bolls are actually part of a hypersensitive response to a bacterial infection from a bacterium called Agrobacterium. Which Professor Brad Goodner studies. So if there's damage to the plant's bark, let's say from an animal or something, Agrobacterium can get in here and start to infect the plant cells. If they're not dealt with, they will eventually spread through the whole plant and kill it. But once the Agrobacterium are detected, the plant cells immediately around the infection start to produce auxin. And what auxin does is it causes hypergrowth of the plant cells right around this area. And so ultimately what you get is you get this kind of bowl, which is wood all around here, which contains all the agrobacterium so they can't get into the plant. And it causes this kind of blob of wood. The infective agrobacterium are kind of stuck in the middle. They can't get through the rest of the wood. Because this is an emergency response, the wood growth is kind of random and rather chaotic. And so people like woodworkers, like to use bowls because there's really interesting wood grain patterns in them. So you can take them and you can carve them and turn them into bowls or something like that. And they have this really interesting wood grain patterns. And it's a response to a hypersensitive response to a disease in a particular tree. Plants can also have what's called a systemic acquired response, which is a plant-wide response. This is much more similar to sort of an immune response, right, where we would create antibodies that would go through our bodies to attack something. The bowl is maybe closer to like what a scab would do, kind of wall off the vulnerable area. whereas the systemic response would be the whole tree. And in some cases, trees can communicate, or plants can communicate, so one plant activating its response against a particular pathogen will spread to neighboring plants, and so they will also activate their response. And hopefully, if they can activate their response before they get infected, they can prevent infection entirely. So there are lots of different ways that actually plants communicate, and there's some really, really neat science about how this works, and how things have evolved in some cases to be so specific, and yet so effective, right? That's why we kind of have to be cautious about phenomena like climate change, where certain species can no longer survive in what used to be their native habitat. So, right, assuming, let's say, the parasitoid wasps die out or maybe move, this plant can no longer resist the attack of the caterpillar, does lots of damage to the plant. Let's say in some cases the plant will die from that damage. So if you remove one small part of this pathway, the whole thing's going to fall apart. And we see a lot of that in sort of evolved responses. Things have evolved a very, very tight balance, and you disrupt one part of that balance, you can throw the whole thing off. So it's not just the wasp that suffers, it's the plant as well. So those things are kind of things that we have to be concerned about. So that's really about plant communication. So there is a quiz this weekend, opens right after class today, closes before class on Monday. It's going to cover sensory and motor systems, animal behavior, and today